Hello dear students, as you are all aware that a wall is made up of individual units called bricks. Similarly, the bodies of all living organisms are also made up of individual units called cells. Cell is the basic structural and functional unit of life. That means our body is also made up of cells? Yes, our body is also made up of uncountable number of cells. Then why are we not able to see these cells? Can we see them? No, we cannot see the cells with naked eye. But yes, we can see the cells by making temporary mount. Today, we are going to make a temporary mount of onion peel and study the plant cell structure. Let us see what all we require to make a temporary mount of onion peel. We require an onion. Why do we take onion? Because we can easily take its peel out. The peel of other plants are difficult to take out and are thick. Pieces of onion which have been kept in water. A dropper. Filter paper, another watch glass, a beaker filled with water, a dissecting needle, a forceps and a brush. We would require a slide for making the temporary mount. Make sure that you hold the slide by its edges, otherwise it can get dirty, and take only clean slide. We also require cover slips for covering the mount which we will be preparing today. How can we see the cells of a plant clearly? Plant cell has an outer tough cell wall which is made up of cellulose. This cellulose cell wall gets easily stained with a stain safranin. Safranin is a stain which gives pink or red color to the cellulose cell wall of the plant. For observing the plant cell structure in detail, we will be using microscope. Would you like to know how a compound microscope works? All of you must have seen a magnifying glass. A magnifying glass magnifies or makes the image bigger of the object which we observe. The same way, even in the microscope, the plant cell structure appears magnified and we can easily observe it. Now, let us see the procedure of making the temporary mount. First, take a watch glass and add a drop of water to it. Now, add a drop of safranin stain to the drop of water. Make sure to cover the stain bottle after you have used it. Now, take a piece of onion and with the help of forceps, take out its peel. This peel should be kept in the mixture of stain and water for about 1 to 2 minutes. Now, take a clean slide and put a drop of water in its center. With the help of the brush and the forceps, transfer the peel onto the slide. While transferring, please take care that you do not fold the peel. Now, take a cover slip and slowly, with the help of a dissecting needle, place it over the temporary mount so that there are no air bubbles in the slide. Take a filter paper and wipe the extra stain from the corners. Our temporary mount is ready for observation. Now, we will place it under the microscope and observe it. What do you observe when you see the temporary mount under the microscope? In a plant cell, safranin stain colors the outer tough wall, cell wall, which is clearly visible. There is a nucleus which is present in the plant cell, which is not in the center, but towards the side. Also, the cytoplasm fills up the entire space of the cell. How do we identify and confirm that the cell that we are observing is a plant cell? Plant cells have a tough outer cell wall. Their nucleus is not in the center, but towards the side, and they are very tightly packed without any intercellular space between them. So, today we have learned the procedure of making a temporary mount of onion peel and also how we can observe and study the cell structure of a plant cell. I hope, dear students, you will be able to follow the process and make the temporary mount on your own. Thank you.